I'm Francis Collins, the director of NIH. I'm happy to welcome you to a Wednesday afternoon lecture, and a very special one, if I do say so myself, and I might be just a little biased here. The title is Fueling the Next Genomic Revolution, Maximizing the Impact of Bacterial, Human, and Human Metagenome, Genomic Knowledge and Technology. That's quite a mouthful, but you're going to see what that all means here in the next hour and a half, because we have a very special lineup here of experts to speak to you. Today, we are indeed celebrating and commemorating some anniversaries. And it happens that 2021 has a series of those that sort of fall in this year. And we genome people have been accused of looking for reasons to celebrate almost everything, including the complete sequence of the human genome, which we have celebrated at least 5 times, and we're probably not done yet. But let's, particularly for today, the 25-year anniversary of the first complete bacterial genome, that being Haemophilus influenzae. And actually, I looked, it was really July of 95, but that's close enough to being at 25 years from now. It is, in a big way, the 20th anniversary of the publication of the human genome from the International Human Genome Project leadership, and at the same time, a publication from Celera of the human genome. These are in Nature and Science, respectively. And 15 years ago, what many people are considering the first human metagenome from Jeff Gordon in Science. Looking back, I could add one more anniversary, and that is the coining of the word genomics. Many of you who have used that almost in every paragraph in your research efforts may be surprised to know that that word only got put out there in 1986. So this is another anniversary, 35th. For the word. So we brought together an all-star team of genomic pioneers to help us, Doctors Claire Fraser, Charles Rojamie, and Eric Lander, and I will introduce them when we get to their presentation. But I wanted to take a few moments here just by way of introduction to set the stage. It was about 1987 when I first heard about the plan to sequence the human genome. I was an assistant professor at the University of Michigan, trying to find genes that caused human disease, and finding it was really hard because there was no sequence, there was no map. Most of the genome was dark matter. We could barely find our way around. Just searching for the cystic fibrosis gene took multiple groups over many years until finally finding a just 3 base pair deletion that was responsible for the most common causes of cystic fibrosis on chromosome 7. But it was brutal getting to that answer. And it was pretty clear that was not going to be replicatable for other diseases that were even less common and maybe less well-behaved genetically. So when I heard about the Genome Project, I thought, oh yeah, please, wouldn't it be great to get this done? Because then this kind of disease gene search could be a lot more efficient. But it also seemed wildly ambitious. Fortunately, there were visionaries, and there was Congress who came up with some money, and there was Jim Watson, who was asked to come and lead the effort at NIH, and it got this off the ground, and a bunch of us got pretty fired up about it, including some of the people you're going to hear about from today. And then Watson was not there anymore, and I got this call from Bernadine Healy, who was at that time the NIH director, asking if I would consider coming to NIH to pick up the reins in this very early stage of the Genome Project, when most people were skeptical at best and actually opposed in many situations. And I said, that's really nice, but I don't think that's what I'm supposed to be doing with my life. And so I said, no, the first time. By the way, you might wonder, okay, Watson had left and Healy was trying to recruit somebody to come and take the reins. Who was running the Genome Project at that point? This is a good trivia question. The answer, our own Michael Gottesman, who's, of course, our Deputy Director for Intramural Research, was for a year the acting director of what was then the National Center for Human Genome Research. Well, ultimately, I realized that this was just too amazing an opportunity to pass up. This was historic. It was going to change everything. Yeah, it might fail, but if it succeeded, why would you not want to have the chance to try to stand at the helm and lead this enterprise and work with all these amazing people? And more of them got on board. And yes, milestones started to happen. Genetic maps, physical maps. Yes, that bacterial genome, which made us believe, hey, you can actually put one of these together. Not long after that, the yeast genome, the C. elegans genome, the first human chromosome, And we had to get to 1,000 base pairs a second if we were really going to do the human genome with its 3 billion base pairs. But that all kind of came together because of a lot of incredibly gifted, talented, and hardworking people. A draft announced on June 26th, 2000 in the White House, but maybe the scientific milestone was the publication in early 2001 of the first draft of the genome with a lot of analysis that many of us spent many hours poring over in various locations, and an experience I shall not forget. So that was one of the reasons, I guess we can say, this is a celebration of a particular moment, 20 years from that. But my gosh, since then, rapid technological advances, transforming our sequencing capacity, increasing the speed, lowering the cost, to now where a human genome is roughly for maybe $500 or $600, where it used to be 400 million. And lots of other applications beyond DNA and the genome, the ability to be able to sequence nucleic acids, including RNA, transcriptomes. And yes, RNA viruses. I have to say one thing about that. After all, it was 1 year ago this past few days, January 10th, where Chinese scientists placed on the internet the sequence of a virus that seemed to be causing trouble in Wuhan. 3 days later, on January 13th, our own vaccine research center decided on the strategy for the vaccine that has now, with the partnership of a company called Moderna, turned into one of the vaccines that has received FDA emergency use authorization. And right at this very moment, NIHers are getting immunized with that vaccine in the B1 cafeteria. And a big shout out then to Fauci and Mascola and Graham and Kizzmekia Corbett, who made all of those things happen. And I don't think it's too much of a stretch to say there is a direct line from what the genomic approach to understanding life was that the Human Genome Project took on, to being able to do something like this at this speed. And now to be able to track all those new variants that are emerging and causing us some concern by sequencing lots and lots of viral genomes for SARS-CoV-2. Well, okay, I divert myself. Of the other exciting possibilities, I'm sure we'll hear about many of them. I will tell you, the basic science of understanding life has just been transformed by the availability of genomics, single cell omics, both looking at transcripts and looking at chromatin structure has just phenomenally changed our view of what's going on in all sorts of human tissues and other organisms as well. The whole metagenomic approach, which we're going to hear more about, become possible because sequencing is so fast and so cheap. Applications, of course, cancer has to be at the top of that list. Almost anyone who now develops a malignancy, you will want to know exactly how that set of cancer cells has acquired mutations in DNA that may be driving the malignancy so that you can choose appropriately what the intervention should be. Newborns now with questionable circumstances, not sure what the diagnosis is. Sequencing the genome has almost become standard of care because you can often make a diagnosis that way more quickly than otherwise would be possible. And in many instances, that may lead to an important intervention, a therapy that you wouldn't have thought of otherwise. And of course, we've now brought this forward into much larger scale in terms of the ability to look at millions of genomes. Certainly a particular million genome that we're looking forward to seeing is the All of Us program, which will be following 1 million very diverse individuals in the United States in the coming years as the largest cohort study that NIH has ever mounted, not just focused on genomics. This is not just another genome project, but genomics is in there along with a lot of study of human behavior and environment and diet and exercise and socioeconomic status, and everything that fits into health and illness, but made possible in terms of the omics part of it by all of these advances. And then, of course, we're getting closer and closer to the point where our ability to look at the genome also leads us to therapeutic options. The availability of gene editing, which is a relatively new event on the scene, beginning to cause us to dream about in vivo applications of gene editing for almost any genetic disease for which we know the specific mutation. My own lab just last week in Nature published a paper with David Liu of the Broad Institute and Jonathan Brown of Vanderbilt, basically in an animal model of progeria, a dramatic form of premature aging, able to show that a single intravenous infusion of a gene editing apparatus targeted specifically to a single letter in the genome, a T that needs to be reverted back to its wild type sequence of a C, resulted in life extension of that animal, which is a pretty good model of the human disease, by almost threefold, and the most significantly dramatic result I've ever seen of a gene therapy intervention, and obviously much hopes, therefore, that that could be extrapolated to humans as well. Well, we have to continue to work to make sure these technologies advance and that they're accessible, and I hope we'll also keep our eyes focused on the importance of applying this across the world, and I'm sure Charles Rojamie will talk about that. But not just the people we study, but the people doing the study also need to be of the most diverse sorts, since we're talking about our shared inheritance of the human genome. So with that little bit of introduction, and I should not go on longer because I will take away the time from the people that you really came to hear, let me stop at this point and introduce our first real speaker, which is Dr. Claire Fraser. Claire got her PhD at State University of New York at Buffalo. She was then on the faculty at Roswell Park Cancer Institute, and then was at NIH from 1985 to 1992 at NINDS, and then at NIAAA, became a section chief, and then moved over to the Institute for Genomic Research, TIGER, where she was and became president and director from 98 to 2007. Since then, she has been at the University of Maryland, where she is the Dean's Endowed Professor and Director for the Institute of Genome Sciences at the University of Maryland School of Medicine. She is an elected member of the National Academy of Medicine. She has, I guess, just about finished her term as president of AAAS and chair of the board of directors, which she's about to become. She's a wonderful friend to many of us. She has been a wonderful person in terms of willingness to help NIH with all kinds of advice and review processes that we depend on experts for. And it is a personal delight to be able to introduce her to you here as part of this special Genomic Symposium. Dr. Fraser, please proceed.
Thank you so much, Francis. I am absolutely delighted to be here. Just want to confirm that you can see my slides before I launch in. Yes?
Yes.
Okay, great. Thank you. Well, I really have the pleasure today of representing two of the domains of life, the bacteria and the archaea. And we'll have the opportunity to speak about how genomics has absolutely transformed our understanding of microbial life on Earth. I'm going to put my comments in a bit of a historical perspective. I think that's important because for students and postdocs who are maybe new to the genomics field, I always think it's important to remind them how far we have come. And I think this historical perspective, which I assure you will be brief, will also showcase how much has been accomplished in the past 25 years in the field of microbial genomics. For me and a number of colleagues, this is where it all began. At the Institute for Genomic Research in 1993 in warehouse space in Gaithersburg. At the time, we were one of the largest DNA sequencing facilities in the world with 20 applied biosystems, 373 sequencers. Most of the effort was focused on human cDNA sequencing. And in our first full year of operation, TIGER completed the sequencing of just over 101,000 samples, which represented a heroic effort. But we really shifted gears in 1994, in large part due to a serendipitous meeting between Craig Venter and Hamilton Smith at an ethics meeting in Spain. Ham Smith, who was at Hopkins at the time, had been working on Haemophilus influenzae his entire career, put forward the idea that perhaps we could deploy the TIGER sequencing and analysis pipeline to see if we could actually sequence an entire bacterial chromosome using whole genome shotgun sequencing. The idea here was not to do any mapping, but just to fragment the genome into pieces of known size, sequence both ends of each of these plasmid and cosmid clones, and then hope that you had an assembly algorithm that would put it all back together again. So we started this somewhat as really a flyer. Nobody was convinced that this was going to work. But as Dr. Collins said in his introduction, the first complete sequence of a bacterial species, a free living organism, was published in Science in July of 1985. This was a project led by Rob Fleischman. It's really remarkable to me to look back at what this entailed. It cost about $750,000 for one bacterial genome sequence, took about 13 months, and the effort of about 40 people. Our assembly represented just over 24,000 DNA fragments, but after enormous efforts in finishing and closure, we did end up with a single circular chromosome, just over 1.8 million base pairs in size. Predicted number of open reading frames, just under 1,800. But I think for all of us, one of the biggest surprises, besides the fact that we were able to accomplish this project, was that 32% of the predicted open reading frames represented proteins of unknown function. This was not at all what we were expecting, but we took a step back and said, well, this is by no means a minimal organism, so maybe we shouldn't have expected that we would be able to place every gene into a known biochemical pathway. So the next project that we undertook in the microbial genomics space with funding from the Department of Energy was the sequence of Mycoplasma genitalium, truly a minimal bacterial species. It had been estimated based on pulse field gel work that the gene size and the number of genes was probably on the order of about 470 genes, and those estimates were remarkably accurate. This was accomplished much more quickly. We had everything in place that we needed, and this work was published late in October, later in 1995. And again, to our complete and utter surprise, about 30% of the predicted open reading frames in Mycoplasma were novel. And I think that at that point, we all decided that we had to accept that this was a fact and that there was a lot of biology that we didn't understand, in this case, even of a minimal organism. And I think we all have seen that this has been a recurring theme in all of the genome projects for the past 25 years. With the two successes in the literature, all of the major funding agencies in the United States quickly jumped on the microbial genomics sequencing analysis bandwagon. The Department of Energy started out with the Microbial Genomics Initiative, and that has, over a number of iterations, morphed into the JGI out in Walnut Creek, California. The NIH, particularly NIAID, initially funded multiple individual pathogen genome projects, and they have now for almost 20 years funded genome centers or genome sequencing centers for infectious diseases. NSF and USDA also got heavily involved and focused on pathogens of interest and relevant to each of their missions. And I think this really speaks to the fact that bacteria are everywhere and they impact life in lots of different ways. I think if you asked any of us, even the most optimistic back in 1995, where this would go, never would we have imagined where we are today. This is information that I pulled recently from the Genomes Online database at JGI. This tries to keep track of all of the projects that are underway, looking at genomes, metagenomes, metatranscriptomes, numbers of organisms that have been targeted with genome sequencing. That number is well over 400,000 now. And this is really a big deal because initially, there was effort underway not to duplicate sequencing effort on strains of the same organism. And this was because cost in the early days was such a prohibitive factor. But it's been extraordinarily gratifying to see where this has gone as someone who was there at the beginning. One of the things that we have clearly seen is that comparative genomics has been a very powerful tool in giving us insights about microbial diversity and evolution. Processes like genome reduction, rearrangement, gene duplication are critically important, but I think one of the biggest surprises, again, that came out of comparative approach was the finding that lateral gene transfer of genes or sets of genes or operons, not only within the bacterial domain, but between the bacterial and archaeal domains of life, was probably an important driving force in generating microbial diversity. I think another really important concept that came out of these comparative studies, particularly when we were looking at multiple isolates of the same species, was what became known as the Pan Genome Concept. There were a number of early studies which demonstrated extensive genetic diversity in isolates of the same species. In some cases, and this first examples was with E. coli, depending upon what strain you were working with, 20 to 35% of the genes in a given E. coli strain appeared to be unique to that strain. The idea that emerged was that dispensable and strain-specific genes probably were involved in facilitating adaptations to various niches. I think a really important concept was that the number of dispensable and strain-specific genes for any given species may greatly outnumber the size of the core genome. And it became clear that it would never be possible to describe a particular species with a single genome sequence. One of the other, I think, really important concepts that emerged from the Pan Genome notion was the idea that you could use this kind of information, looking at genes that are shared versus genes that are unique, in the field of reverse vaccinology. This was something that was coined by Rino Rapulli and his colleagues at Chiron Vaccines, a group at Tiger. Still has a very long-standing collaboration with Rino and his colleagues. And the idea here is really very simple. You don't have to start by growing an organism in culture. You can start with a genome sequence, look to identify potential vaccine candidates. Here in panel B, these would presumably be candidates that might be secreted or be found in the outer membrane or the periplasmic space or the inner membrane. You could then take each of these candidates individually, purify them, immunize animals in preclinical studies, do serological analysis to identify the most robust vaccine candidates, and then look to see what the sequence conservation might be, with the idea being that you want to try and find potential vaccine candidates that are highly conserved, which would increase the range for a given vaccine. We put this idea to a test with this first project on Neisseria meningitidis, serogroup B, again, a Tiger Chiron collaboration. And about 4 years after completing the genome sequence of MEN-B, vaccine candidates entered clinical trials. And a number of years later, after various iterations and improvements in a cocktail of MEN-B antigens, this vaccine, Bexsero, made it to market from GSK. And this is the first vaccine that was developed solely using the reverse vaccinology approach, another accomplishment that we are very proud of. Microbial genomics has played a key role in helping to develop the field of microbial forensics. Jacques Ravel, Dave Rasco, and I at Tiger spent 7 years working with the FBI on the Amerithrax investigation. And what we were able to ultimately do is to use complete genome analysis to demonstrate that there were mutations in these various morphotypes of Bacillus anthracis that had been recovered from the material sent through the mail. These genome differences allowed for the development of PCR-based assays that were used to screen over 1,100 anthrax stocks from labs around the world, and provided some very robust evidence, I think, that pointed to a potential source of the material as part of that was the material for this anthrax mailing. Microbial genomics has also played important roles in surveillance and outbreak tracking. By no means is this meant to be a complete list, but this has really become the way in which outbreaks are tracked because it gives now quick information and information with exquisite sensitivity. I think for me, one of the most exciting developments in the entire field was when we moved from looking at single isolates that you had to grow in culture into metagenomics beyond the single genome here. In a key paper published in 2004, Joe Handelsman made a very compelling point that if we really want to understand microbes, we need to be looking at them in their natural environments because that's where they live. They don't live in isolation. We could think about this now in 2004 because DNA sequencing was no longer the bottleneck, either in terms of throughput or cost. And one of the keys here was this meant that we could now take a culture-independent approach to studying microbes. And we knew from lots of previous work that the uncultured microbial world was far greater than what could be grown in culture. What metagenomics requires is one to take more of an ecosystems perspective in looking at and trying to decipher microbial communities. And this ultimately generates a multidimensional picture of community structure and function. I think this is the paper that Dr. Collins referred to in his introduction. This was a paper published in 2006. It was a collaboration between the Tiger Group, Jeff Gordon, and David Relman's lab, looking at doing the first metagenomic analysis of the human distal gut microbiome in 2 individuals. This was an important paper, I think, because it demonstrated this could be done. We identified key bacterial and archaeal species, found evidence for strain variation. We were able to reconstruct a number of metabolic pathways from the metagenome data, and we speculated that the human microbiome data would be of value in better understanding human health and disease. And I think that has indeed turned out to be the case. NIH jumped on this and starting in 2008, and I remember being at some of the planning meetings with Dr. Collins, but in 2008, the NIH Human Microbiome Project was launched. Second round of funding was the Human Microbiome Project, the integrated HMP. This was very exciting for me because this really meant that microbes entered the NHGRI research portfolio. They were no longer just in the domain of NIAID. And the work that has come in the last 12 years or so has been truly astounding. We have come to understand that to really understand these communities, we need multiple omics to truly understand function, both DNA, RNA, protein, and metabolite-based approaches. And the real power comes from integrating all of these different data sets. I think we have come to really understand that when one thinks about the human microbiome, it truly is an ecosystem, and many factors, both intrinsic and extrinsic to both the microbiome, the host, and the environment have been identified that have profound effects on modulating the composition and function of the human microbiome over the course of a lifetime. One of the important ideas to come out of this is that there is such a thing as a healthy microbiota, a healthy microbiome. This is more defined in terms of function, not in terms of composition. And on the other side of this is the notion of dysbiosis, that disease, environmental factors, other things can shift the balance from a healthy microbiota to one that is dysbiotic, and that is often associated with a number of diseases. This idea led Philippe Sansetti and colleagues to propose an interesting new twist on Coax postulates, the ecological Coax postulate. The idea being that if this is indeed the case, that dysbiotic microbiota are associated with disease, that one should then be able, through fecal microbiota transplant as an example, put a dysbiotic microbiota into a healthy animal and recapitulate the disease. And that has really sort of turned the original Coax postulates on its head. Another really exciting development to come out of the microbiome area is the interest that it's generated from the biotech and big pharma industry, both in terms of novel diagnostics, novel therapeutics, and there is a tremendous amount of work underway in this space. Just some examples are shown here on this slide. Another thing that I have found in my career is that the microbiome has generated a tremendous amount of interest on the part of the public. There are a number of citizen science efforts that are underway. The American Gut Project, there's a corresponding British Gut Project. They have transitioned into the Microbiota initiative. There is Your Private Biome that is looking at the vaginal microbiome. But there is just a tremendous interest on the part of the public in understanding the microbiome, and I think understanding what individuals can do to promote a healthy microbiome over the course of a lifetime. And I'm going to just finish up with this last plug for colleagues at the Department of Energy. This was a paper published in November of last year, a genomic catalog of Earth's microbiomes, because I don't think we should forget that in parallel with all of the human work, there is other environmental microbiome work underway. This paper to me represented a real tour de force. Over 52,000 metagenome assembled genomes from diverse habitats were reported on. Included in this data set were over 1,200 novel candidate species, and this data set expanded the known phylogenetic diversity of bacteria and archaea by 44%. And I still think we have a very long way to go before we truly have surveyed the diversity of microbial life on Earth. My last slide, the future awaits. It's always difficult in a field as large as this to speculate where things were going to go, but I think it goes without saying that novel pathogens are out there waiting to be discovered, waiting to wreak havoc. But as was pointed out in the introduction, Genomics has provided such a robust platform for characterizing, identifying, and classifying any novel pathogen that might come along. As I mentioned, I think that we're just in the infancy with regard to new microbiome-based diagnostics and therapeutics, and I think there will always be great interest in carrying out additional surveys of microbial life on Earth. So for me, it has been a tremendous privilege to have been part of all of this for the last 25 years. I don't have an acknowledgement slide because I couldn't possibly acknowledge all of the colleagues and collaborators that I've had the pleasure to work with. But there has been, from a handful of early investigators that were involved in this work, there is now a cadre of really talented microbial genomic scientists. And we're now seeing second and third generations of microbial scientists emerge. So I'm just delighted that this was included in part of this symposium, and I look forward to the discussion. Thank you.
Claire, thank you for a wonderful romp through a lot of microbial genetics in a very short period, and genomics as well. My goodness. Yeah, again, people who are interested in engaging in the discussion, you can submit questions via the webcast website at the Send Live Feedback button just below the video display. But let us go on to the next presenter. We're going to have a panel at the end. So next to speak to you is going to be Dr. Charles Rojamie. He was born in Nigeria, got his undergraduate degree at the University of Benin. Came to the U.S. For a master's degree in Mississippi and then an MPH PhD at the University of Alabama at Birmingham. He then moved to Loyola University Medical Center, which is where I first encountered him. And I had the privilege of working with him on one of the first efforts to try to do genetics of common disease in Africa. That was a study of diabetes in Ghana and Nigeria, which is continuing to this day. But he got recruited to Howard University in 2003 and became director of the Howard National Human Genome Center for the next 5 years. And then came to NIH in our intramural program, where he is NIH Distinguished Investigator and Chief of the Metabolic, Cardiovascular, and Inflammatory Disease branch in NHGRI. He has very recently taken on the role by election of being president of the American Society of Human Genetics. And he is also an elected member of the National Academy of Medicine, the African Academy of Sciences, and he is the founding president of the African Society of Human Genetics. It's been a great privilege to work with Charles on a lot of interesting problems over the course of 20 years. He played a huge role in our effort to diversify the DNA samples in the HapMap projects and in 1,000 Genomes projects, and is now a real leader in pushing forward the importance of paying attention to diversity in everything we do in genomics. So Charles, thank you for being part of the symposium, and please take it away.
Yes, thanks, Francis, for that wonderful introduction. And I'm really excited to be a part of this presentation today. My goal is to share with you why it's important for us to study African genomes, and that by studying African genomes, we're actually studying the genomes of everybody. And hopefully by the end of my presentation, you will see exactly what I mean by that. So the first thing I want to say is that Africa, or the geographical area we call Africa today, is really the ancestral home for all humanity. And modern humans originated in Africa about 300,000 years ago. And about 99% of our human evolutionary experience was spent in Africa after we, the chimpanzee human divergence about 6 million years ago. Again, as we like to travel, we like to see different places, a small group of us, then decided that they wanted to migrate out of Africa to populate the rest of the world about 100,000 years ago. Now, this common history of humanity is really why we share most of our genetic inheritance. And that is very important for us to continue to recognize. So, like I said, a small group of people migrated out of Africa. But what happened was that that small group, when they left, they did not carry all of the genetic variation that existed in Africa at that point, what we call in population genetics, the bottleneck effect. So a subset of the common variants before this migration remain in Africa. And Africa is the only place that we can truly systematically study the importance of these variants. And in our understanding of human history and also our health. So as Francis said during the introduction, I was really, really excited to participate in various genomic projects. One of the motivating factors has always been that we need to make genomics a global effort. So as you will see in subsequent presentation in terms of HapMap and also the 1,000 genome, we're able to show very clearly that African genomes have more variability and carry more diversity, and that by studying this diversity, we can indeed understand human history and human health. Whether we are looking at SNPs, non-synonymal variants of or loss of function variants, you will see that African population indeed do carry more of these variants, which is absolutely important, and I will show you some examples. So there's more haplotype diversity in African genomes, and this diversity of haplotypes and other variants can indeed be used when we are mapping genes, when we are trying to better localize where signals are in our genome. And I'm now going to show you an example of a recent work from our lab that demonstrates this in terms of circulating resistant level, how we took advantage of the short haplotypes and haplotype diversity in African genomes. And I hope this video will behave and play correctly.
In this manuscript, Dr. Charles Rojamie's group at the National Human Genome Research Institute identified the genetic architecture of circulating resistant levels in Africans from Nigeria, Kenya, and Ghana, and African Americans from the Washington, D.C. area. Resistin is a small peptide secreted by bone marrow cells in humans, and it's been suggested as a biomarker for metabolic syndrome and type 2 diabetes. Two previous genome-wide association studies performed in people of European and Asian descent identified significant associations with genetic variants near the RETN gene, that's the gene that encodes resistin. Dr. Rotimi's group performed GWAS for plasma-resistant levels using more than 18 million genetic markers in nearly 4,000 continental Africans and nearly 2,000 African Americans separately. Both GWAS identified the same variant, 75 kilobases upstream of the RETN gene, as the most significantly associated SNP. The haplotype structure in the African populations allowed the researchers to fine-map this locus to a single variant, and whole exome sequencing studies ruled out the contributions of coding variant in the RETN gene. Further, the GTEx dataset demonstrated EQTL effect of the index variant on resistant expression in immune cells. Together, these results suggest that regulatory variants that affect resistant expression levels derive the variation in plasma resistant levels. Finally, the authors performed Mendelian randomization analysis of resistant levels of metabolic phenotypes. However, they did not find evidence of a causal association between resistant levels and metabolic syndrome-related phenotypes. Overall, this well-executed study was able to fine-map the RETN locus for resistant associations, and perhaps more interestingly, cast some doubts on the idea of resistin as a biomarker for type 2 diabetes.
Thank you. So another way we demonstrate, or I try to simplify our understanding of the huge diversity of variation in the African genome is by using this cartoon here, which is the a Yoruba man playing what we call the talking drum, and the Maasai. So if you compare the genomes of these two African individuals, they are more likely to be different from each other than when you take one of them and compare them to this guy, again, which speaks to the huge diversity that exists across Africa. What is the potential implication of this? So in the context of public health, we, myself and Lynn George, in the paper that we wrote about, I think 2010, we used the hypersensitivity to HIV drug cover to demonstrate this very point, that if you look at different African populations, you do see huge differences in the distribution of variants that are very important. In this case, pharmacogenetics, how people respond to drug. Initially, the thinking was because it was about 1% in African Americans that we did not need to screen Africans for this variants that is very important in response to an HIV drug. But when you look at using the HapMap data, we show very clearly that among the Maasais have one of the highest rate, if not the highest rate in the world. And that variance is very rare among Yorubas. So the point here is that by using labels like Africans or Blacks, we can indeed distort our understanding of human genetic variation, and we can indeed arrive at wrong public health decisions. So again, these are all in a way to demonstrate and recognize that we need to study different African populations if we are truly going to deploy precision medicine effectively. So this is a recent study, again, with Mary Claire King and my friend Raj Messer in South Africa, where they demonstrate again the importance of African genomes, in this case, in the context of schizophrenia. And a lot of the studies that have been done in Europeans, but by looking at exomes in the host of people in South Africa, they observed that overall, the genetic architecture of schizophrenia in Africans is generally reflective of what we see in Europeans, but that the greater genetic variation in African provides more power to detect relationship of genes to phenotype. And that what was found in Africans really does provide information for us to truly understand the genetics of schizophrenia in a global context. So for my own research and some of the work that I've done, again, collaboratively with Dr. Collins and others from Howard University and in different African countries, despite the fact that GWAS in Europeans have been extremely successful in identifying loci for type 2 diabetes, recently we were able to discover novel variants in the gene called ZRMB3 in Africans that were specific for type 2 diabetes in Africans. And that study was made, and this finding was made possible by our study of what we call the African American diabetes mellitus, which was actually started as a result of Francis' experience as a physician in Nigeria some years ago. This study now has enrolled over 7,000 individuals from Ghana, Nigeria, and Kenya. And we use that GWAS to find this locus in ZRMB3, which has not been identified in European population. And it turned out that this variant is actually very, very informative in terms of beta cell mass. So when we knock down this variant, we were able to demonstrate very clearly that there's a reduction in beta cell number in zebrafish. That was extremely exciting to us, and we went further to show that when you suppress ZRMB3 in mammalian beta cells, that you do impair insulin secretion. And when you silence ZRMB3, you do show that it will show very clearly that the beta cell response to glucose challenge is significantly diminished, as a result of the silencing of this gene. So this again provide insight into the genetics of type 2 diabetes in African populations. And I do believe that as we continue to understand this biology, that this finding is not just going to be important to African populations. Similarly, we found a variant, again, that was African specific in the Sigma 4D. This is again very, very important because individuals who carry these variants, about 1% in West African and African Americans, actually weigh about 6 pounds heavier than the non-carriers. And we showed that elevated level of Sigma 4D was clearly associated with obesity risk. And in both men and women, individuals who are obese really do have much higher level of Sigma 4D, again demonstrating the importance of these variants in our understanding of adiposity in human populations. And like I said, we could not have found this in European and Asian population because this variant is indeed monomorphic. So similarly, a multi-ancestry study that in collaboration with our colleagues in the charge gene lifestyle interactions, we were able to demonstrate very well that by studying African ancestry populations, we can make novel discovery, especially in the context of gene environment interactions. In this case, lipids and smoking. So by looking at, again, different models of interactions, we were able to identify 13 novel variants. What was actually fascinating is that 9 of these 13 variants were statistically significant in African ancestry populations, or the trans-ancestry analysis that included African individuals. Now, what would be rather obvious is that some of these differences may be due to allele frequency differences between African ancestry individuals and also European individuals, which is indeed true for 7 of them. But for 2, the frequency is actually higher in non-African populations, and still we did not find signal in those in non-African ancestry populations. As you will see for this specific SNP here, you'll see that in non-African populations, indeed, there's really no signal, whereas in all of the different studies showed very strong association of those studies of African ancestry individuals. Interestingly, this particular SNP is an trans-EQTL for a taste receptor that is very important in terms of cigarette smoking. So, although we're excited with this finding, we're even more excited when this study was released, a collaboration between FDA and NIH investigators that identified variants that were specific to African ancestry populations in terms of preference for menthol cigarettes. So for a long time, epidemiologists and public health individuals have known that African Americans who smoke, over 80% of them prefer mentholated cigarettes compared to about 30% in Europeans. And nobody really do understand the reason for that. But this finding in the G coupling protein showed that was associated with a 5 to 8-fold increase also of menthol cigarette smoking, provided insight into this finding because this variant, a particular haplotype, was indeed African specific. And it's important, cell signaling and also influences menthol smoking. One of the things that individuals, the speculation has been that African American prefer, or people who prefer menthol cigarettes do so because it's less irritating. But the downside to that is it's less irritating, but you may indeed encourage you to smoke more. So this is an interesting public health finding that I think should help us to understand the public health issue. Now, a major example, perhaps one of the most exciting recently, is the relationship between variants in the APOL1 gene and kidney disease in African ancestry individuals. In 2010, there was this major publications from NIH, colleagues and Johns Hopkins, using admixture mapping to localize variants that were indeed important for kidney disease. But further studies using sequencing efforts, discovered that the variation was actually in the APOL1 gene, and the odds ratio was, phenomenally high, about 10.5 for end-stage renal disease for individuals who carry 2 copies of these variants. Now, what was absolutely fascinating is that it is very common in West Africans and African Americans, but absent in Europeans and Asians. The APOL1 is a serum factor that lyses trypanosomes. And we believe that it rose to high frequency in this population because of protection against the lethal form of African sleeping sickness. Now, what has this to do with kidney disease? We know from public health and epidemiology, despite the fact that African Americans make up about 13% of the US population, they make up 32% of kidney failure. They are 4 times more likely to develop kidney failure compared to their white counterpart. Now, individuals who have 2 copies of this variant are 4 times, or maybe 40% more likely to experience, to be on dialysis. And again, very, very important, helping us to understand some of the disparity that we see in terms of kidney disease and kidney failure in African Americans. Importantly, there's no way to use skin color or appearance to now know who may carry these variants, because these variants are spread all over the world as a result of slave trade, as a result of individuals sharing DNA across the world. And so it's therefore very important to understand who may indeed carry these variants before individuals donate kidney or before individuals accept kidney from donors. So there needs to be a need for screening for this particular APOL1. And I think global studies are ongoing to truly see how we can implement this in the clinical setting. So again, as Francis said during the introduction, studying African populations and African genome is, as I hope I've shown to you, that is very important in terms of understanding science and human history, but it's also a social justice issue. Because if you look at what we have done so far in genomics, especially what we call genome-wide association studies, you will see that African participation in such effort is almost non-existent. We are beginning to change this, as I will show to you in a minute. But the question becomes, if we do not change course, will tomorrow's medicine work for everybody? I think that's a very important thing that we have to always be conscious of. And as shown, in 2016, I think, where African Americans were being diagnosed with heart disorder mistakenly because the reference that was being used did not have enough variation from African genomes. The same thing is true as we begin to think how we can deploy polygenic risk scores, which is basically using aggregation of variations that we find from genome-wide association study to stratify individuals in terms of their risk to different conditions like heart disease, diabetes, and others. So if we start with European discovery, variants from European studies, we'll see that Africans, when we try to use that in predicting what will happen in Africans, we do very, very poorly. So this means as a pay-head disparity if we don't change course as we deploy polygenic risk scores in genomics. And we have a paper under review now from my group that also has demonstrated that we do worse in African ancestry populations when we start from European populations in terms of additional way we can explain risk, in risk models. So again, showing the importance of studying increasing diversity in genomics. Now, I want to share with you how we have begun to change this, in terms of what we call the H3 Africa, which is Human Hereditary and Health in Africa, co-funded by the Wellcome Trust and the NIH, with a leadership from Francis and significant funding from the common funds, have really now begun to change the course of participation of Africans in genomic studies. So far we've been able to attract over close to 180 million variants, and a lot of investigators and a lot of participants engaged. And a lot of studies have been established: mental health studies, cardiovascular disease, microbiome, infectious disease, and other genetic diseases. So recently, we had a major publications that shared significant insight into the genomics of African people and of the world in general. And we uncovered 3 million variants, that are indeed very, very important. And these were mostly in the ethno-linguistic groups that have not been previously sampled, again demonstrating why we need systematic sampling across Africa. We discovered 62 previously unreported loci under very strong positive selections that are indeed important for viral immunity and DNA repair and metabolism. We demonstrated a very complex pattern of admixture across African populations. Again, very important in the way that we understand different ancestral background in genetics. And we hypothesized a new, route for the major Bantu expansion across Africa. Again, coming from the eastern part of West Africa, through Zambia to South Africa and East Africa, again informing human migrational patterns in that part of the world. And one of the things that was absolutely wonderful is the finding among boron in the middle part of Nigeria, with a very high level of Nilo-Saharan ancestry. And we don't know how that got to middle part of Nigeria, because that's ancestry that is seen mostly in East Africa. So again, indicating that we need to have the systematic sampling. Perhaps medically, this is really, really interesting. Pathogenic variants that have been, in genes that have been characterized as very medically relevant were uncommon in this study. And variants denoted as likely pathogenic in ClinVar were very common. So what does this mean? I believe that we need to study more African populations so we can better classify what is pathogenic and what is not. We identify gene flow, and we show that response to human disease is a strong driver of genome level population variations. Again, all of this informing why we need to continue to study African populations. For being, as we started H3 Africa, one of my points, and I think Francis and others made this very strongly, that it is not only important to include Africans in genomics as participants, we have to increase the number of African scientists that are participating in genomics. I want to say today that I'm extremely happy to show that this major paper in Nature was led mostly by African investigators in Africa and also in the diaspora. So when we're doing HapMap, we recognize that due to the high diversity in African populations, that the GWAS arrays that we're developing were not very, very efficient in integrating African genome. So one of the things that H3 Africa has also delivered on is the development of a new array, a Pan-African array of about 2.5 million that is more efficient in interrogating African genome. And I will encourage all of us that if we are indeed doing genetics of African people in the context of GWAS, to seriously consider using this chip or similar ones. So we continue to evaluate. Again, this was a study that was led by two colleagues in my lab, Bentley and Shaniqua Kala, and we showed very clearly again that we are beginning to make progress, but we are not there yet. I want to highlight some studies here, like TopMed from NHGRI. I believe that TopMed has radically changed the ancestry of individuals that have been sequenced to date. And that is due to a direct effort of encouraging investigators to include African ancestry individuals in genomic studies. Page Consortium and the charge also H3 Africa have indicated, CAPA and NeuroGap, and then recently the All of Us and the program from the NIH, beginning to change the number of African individuals that are included in genomics, which is absolutely wonderful and music for me. So I'm going to end by saying the story of humanity is still being told, especially what we are learning from our other human relatives, Neanderthal and Denisova. And one of the things that is becoming quite clear is that individuals who migrated out of Africa indeed share DNA with Neanderthal, and we have signatures of that in various genomes. And they have medical implications in diabetes, in immunity, and we see even some evidence now in terms of COVID-19, where the ancestral background coming from the Neanderthal introgression is indeed having biological importance. So our story is still being told. Now, the point I want to make here is that we cannot comprehensively tell this story without the systematic sampling of African genomes. And notably, the genomic story of humanity can be told without reaffirming old prejudice. Variations in norm in our genome and diversity is not an illusion. We need to study it, but we need to study it looking at all of us as having a common origin. And the elasticity of our genome has captured the need for us to survive different environmental settings. And that if we truly understand that, we can truly understand genomics in health and history. And I thank you for your time. And I just wanted to say that this is me here, the odd guy out in a sense, pretending to be a warrior that he knows nothing about. Thank you very much.
Thank you, Charles, for a really fascinating romp through a whole lot of really interesting science over a couple of decades. And we are running a little late, so I should quickly move on to introduce the next presenter. Well known to many of you, Dr. Eric Lander got his undergraduate degree at Princeton, PhD in mathematics at Oxford. He was a Rhodes Scholar. Went on to be a professor at Harvard in the School of Business, interesting, and then to MIT in biology, and then became director of the Whitehead, and was a leading force, to put it mildly, in the success of the Human Genome Project, where I got to know Eric really well and learned to be amazed at what he could accomplish and his ability to think both quantitatively and at scale. He is now professor of the MIT Department of Biology. He's president and founding director of the Broad Institute and professor at Harvard Medical School. He's an elected member of both the IOM and the National Academy of Sciences. And he was co-chair of the President's Council of Advisors on Science and Technology, PCAST, under President Obama. Has received both the Breakthrough Prize in Life Sciences and the Albany Prize in Medicine, and is always an interesting person to hear from about genomics. So I can't wait to see what he's going to throw at us today. Go ahead, Eric, toss it out.
All right. Well, Francis, thank you so much for the introduction. And it's a tremendous honor to follow Claire Fraser and Charles Rojamie. First, a declaration of personal and institutional interests. And so I will then go on to, yes. What I'd like to do today is take the closing minutes and talk about the future. The past has been a lot of fun. It's been amazing to watch what has gotten done now over the course of, well, Francis has talked about anniversaries 35 years ago and 25 years ago and et cetera. But my sense is there's more future than past. And so I really now want to turn to the questions of where do I think human genetics in particular is going? And so, I'll briefly take a romp through what we might call phase one, 1980, all the way to maybe 5 years ago. And these dates are, of course, slightly arbitrary, but not entirely. 1980 was when David Botstein and colleagues published this very elegant, important paper that said we could map genes using genetic markers. Well, they knew that in fruit flies, but they didn't have a way to do it for an organism like humans, and DNA polymorphism provided those genetic markers. And at the time, we thought about it like simple fruit fly crosses, but we never appreciated back then how powerful it could get. And David dragged me into the field of human genetics by asking questions about, well, what can we do for things that weren't caused by a single gene? And eventually, these sorts of ideas have led us all to being able to map highly polygenic diseases. But before either of these two things could be done, we did actually need to have a map of the human genome and a sequence of the human genome. And Francis and I, and maybe about, several thousand more people devoted themselves to the work of trying to get that information and make sure it's totally freely available to everybody. And one of the anniversaries we're celebrating is that. But when the one human genome or so were done, we realized that was just one. And if we really wanted to be able to study human genetic variation, and particularly in complex diseases, as soon as we were done with one, we had to think about such crazy things as finding all the variation in the human population. You know, we would want a catalog of all common genetic variation in the population. Common is a somewhat arbitrary term. At the time, we were thinking down to 1%, and then 0.1% and 0.100%. We'd want to figure out how it was correlated with each other, the so-called haplotype structure of the genome. And then if we were going to put it to any use, we were going to need to be able to genotype, tens to hundreds of thousands of polymorphic markers in tens to hundreds or thousands or millions of samples. And we need to be able to analyze all of that. And this was certifiably insane in the late 1990s when it was being thought about. But as with everything we have learned with genomics, if you apply your mind to it, you can make great progress. And by 1998, we had very little, and we were genotyping one at a time. And by 2007, not even a decade later, we had 10 million polymorphisms across the genome. We knew how they were correlated, and we could genotype 1 million at a time. And the result of that has been this mapping of genetic loci that underlie common genetic diseases. Not the 2 or 3 or 4 genes people might have thought of for type 2 diabetes, but 400 and 160, and it's up to more for coronary artery disease and schizophrenia using GWAS, or common variant association studies. And as sequencing has come along, we've got rare variant association studies that are finding strong loss of function mutations. So now we have something like 100,000 associations between genetic variation and particular traits. And sometimes it's possible to go from that genetic association, meaning that this variant is somewhat enriched in people with the disease, to pin down the gene that is involved in it. Each of these has been a beautiful but heroic scientific effort. For example, to figure out that sickle cell is modified by a gene that affects fetal hemoglobin levels or other things. And it's provided great insights into some diseases, autophagy and inflammatory bowel disease, showing that HDL, while epidemiologically associated with protection from heart disease, is not causally associated. That in schizophrenia, a set of genes involved in synaptic pruning play a role, pointing to a process that had not been specifically implicated, and the role of microglia in Alzheimer's had not been appreciated till genetics pointed us at that direction, because I think, maybe not unreasonable, people thought Alzheimer's neurons, but the genes thought otherwise. So phase 1 has gotten us thousands of disease genes. Maybe it's 6,000. I've stopped trying to update, but essentially any Mendelian disorder for which we have enough cases, the gene can be wrestled down to the ground by a student. For common disease, these more than 100,000 loci establishing the polygenic basis of disease. And what we learned from the Human Genome Project to today is that there are secrets to success of this. Every one of these things required clear scientific vision, the willingness to take comprehensive views of biology, develop powerful experimental tools, data platforms, data sharing. One of the great contributions of the Human Genome Project was this total commitment to data sharing, that if you want to get something done, pick some flagship projects that rally people around it, and international collaboration, worth reminding ourselves of the power of doing that. And as I say, many basic ideas are now clear about the genetic architecture of disease. But for these 100,000 loci that have been mapped, it's still too much work to find the individual genes. And it's got to be less than 1% of the loci for which, 1% of the associations for which we have definitive evidence of the genes that are involved. For some diseases, we have reasonable guesses for 20 or 30% of them, but no way to do business. We really, really want to be able to go from maps to mechanisms to medicine, which is the watchword of this international collaboration called the International Common Disease Alliance. And I think this is phase 2. To be able to reliably go from samples to variants to genes to cell types to processes to disease reliably, not through heroic efforts, but like falling off a log, like we can map Mendelian disorders today. Now, why have a bunch of scientists from around the world, really hundreds now, rallied around this goal? Well, as always, it's because there are powerful scientific forces and powerful scientific tools that are driving it, making it possible. And the 3 big ones to acknowledge are large-scale human cohorts. You can't do this with a genome or 100 genomes, but you can do this with large biobanks, such as the UK created in Estonia and Finland, and most excitingly, the United States with the 1 million person All of Us cohort. And increasingly, if we are able to make use of medical records with patients' consent and protection of privacy, we can begin to hallucinate being able to do genomic analysis on tens to hundreds of millions of people. Of course, to do that, we got to be able to analyze genomes quickly and cheaply. And that's what's exciting. As Francis said, costs of genomic sequencing are dropping $500 or $600 today per genome. I suspect it'll get down in the neighborhood of $100. And well, who knows how low it can drop, but maybe it'll drop lower than the cost of drawing a blood sample or something. In any case, I think you'll see that this gets used in the course of medicine routinely. But it's not just whole genomes, it's the really powerful single cell analysis, single cell RNA sequencing, single cell chromatin analysis at massive scales. We're already up to 100 million cells. Billions of cells is well within range, and much, much more than that. And then the genomic technology of being able to edit genomes. If you have an idea of what is the particular role of a variant, well, you can just change it and see if you have the right kinds of high dimensional readouts. And needless to say, this would never work without massive cloud-based data platforms and technologies to ensure federated, secure data ecosystems. So I'm just going to quickly touch base, because I know we don't have much time, on some of these key points. Human populations, human cohorts have been surprisingly powerful. I don't think any of us, when this was started, imagined what this would turn into. But you can take any disease or any phenotype and map dozens to hundreds of loci for disease progression phenotypes, treatment responses, family history, development, and, by the way, an international collaboration that is going on so quickly and was set up just in the last year on COVID-19 susceptibility. But if you really want to take the learnings from the whole human population, you have to really celebrate the amazing range of genetic variation we have. Isolated populations like Finland or different parts of India have the natural ability to reveal stronger alleles. Populations that have consanguineous marriages have the possibility to reveal recessively acting alleles in ways that we can't see, say, in my home of New York City. And Africa, as Charles Rojamie has said, has amazing power for genetics because, for just to pick one example, it has an ability to do fine mapping of causal variants by taking advantage of the amazing diversity and shorter haplotypes there. And so no part of the world is an island for human genetics. If we're going to learn together. Now, by looking at these human cohorts, we can also discover other things. Having hundreds of loci that show association or 1 million markers across the genome, we can begin to build polygenic predictors that say who's at higher risk of disease. And Charles gave a very good example of that. And what's remarkable is we've now hit the point where the people at the top of the polygenic spectrum really do have meaningfully higher risk. Say threefold higher risk might be present in the top 2, 3, 4, 5%. And that's a point where you can imagine that in clinical care, some people might be referred for statins at a lower age or mammography at a different age. And so all of this information from polygenic risk scores is very different than we thought we might have, and it's appearing for common diseases. Other ways in which this information is so surprising, you can, by just looking at the association of millions of markers across the genome, you can begin to, and across hundreds of different traits, you can discover the biology of a locus. Pick a locus like SLC39A8 that includes a manganese transporter and look at all the diseases it's associated with. And you can begin to pick up about the potential biology of that. And a reasonable good guess from this is it affects glycosylation in a variety of different ways. You can look across phenotypes, different phenotypes for psychiatric disease, and say of the 1 million markers across the genome, if I run a correlation between schizophrenia and autism, do I see a correlation? That is, are there common modules underlying it? You can intersect the genetic markers associated with a disease with the gene expression patterns in given cell types to figure out what cell types might play a role in the disease and what genetic pathways, what cellular pathways might play a role. Now, we are going to see, I'm sure, an explosion in the number of genomes that are available. People make projections. I'm not going to stand by any projection. I took one from the literature. But if you think genomes at $100 as a target, and humans, if we round up, living 100 years, the amortized cost is about $1 per year to have that in your medical record. And that's like a lot cheaper than annual cost of healthcare in the vast majority of at least developed countries in the world. And so we ought to be thinking that collecting this information should be and will become routine. And by the way, that's a really good thing because we're going to need that. Right now, when we go off looking for an association study with type 2 diabetes or Alzheimer's, you have to collect patients with type 2 diabetes or Alzheimer's. But if the databases, the biobanks get big enough, there will be enough cases in the biobank. No, the 1 million person, all of us cohort won't be enough for everything, but I'm pretty confident that the 1 million person, all of us cohort is not going to stop at 1 million persons. It's going to end up being much better. And I view all of us at 1 million persons as a pilot for what we really want to do, which is to be able to turn the healthcare system into a learning system with the cooperation of all those patients who do want to contribute to that in a secure way. Now, I want to really emphasize Charles's point just one more time, and here I'll point to work of Alicia Martin on the incredible importance of increasing the diversity. We have such a huge diversity problem. It's great that we have lots of European samples, and it's unacceptable that we do not have comparable numbers of samples from other parts of the world. And it's unacceptable because, as I've already indicated, there's so much we can't learn without it. And it's also unacceptable with regard to these polygenic risk scores, because a polygenic risk score created with European data is optimized for European patients. Just because of the genetic linkage relationships and fluctuations and things, and it performs less and less well as we move to different populations, and especially poorly for an African population, but also poorly for an East Asian population. And we should just decide we are going to be serious about collecting biobanks from around the world. And as has already been noted, it has to be done led by the people in those countries, because we need not just diversity in samples, we need diversity in the investigators who are doing this. I'm going to take a quick moment and I will not spend time on this because I want to stop to keep us on time, but it isn't just human genetic studies. The real power now is also becoming understanding gene regulation, because most of the polymorphism that we have available to look at is polymorphism in non-coding regions. And so as this little picture indicates, we have got to associate lots of non-coding variation with the target genes. And it can be done by different ways: observation, perturbation, biochemical rules, and prediction. We can get these data about gene expression and chromatin state and active enhancers across the whole genome. And we need to do it. And the Human Cell Atlas is leading the way for every cell type and for lots of people. We'll also need to do this if we want to believe in causality by perturbation, being able to use tools like CRISPR and massively parallel reporter assays and assays in cell lines and organisms and primary tissues. And I've put in parentheses a favorite crazy idea I have of stochastic fluctuation, even in supposedly cells at steady state. We're going to need to actually convert them to biochemical rules, because we're not going to measure each experimentally. And so work, for example, of my former student Jesse Engreit's to build predictors, I think will be an important part of this, as will these powerful tools of artificial intelligence. So I'll skip on simply to say data will be a critical part of it. The model has been inverted. The days when you used to download a data set to work on it are over. The data sets are much too big. We have to have the data available to all of the researchers, and we need to have available data platforms so that people can use the same tools on many different data sets, have them open and freely available. And there are many efforts around the world now to do it. And I am excited that they will produce platforms that will allow students anywhere, yes, at MIT, but in Kansas and Uzbekistan and in South Africa and in Nigeria, to be able to analyze these data. So the International Common Disease Alliance is something that I've gotten involved in helping to start, but together with a lot of other people. There's an organizing committee of 35 people, including the NIH's own Carolyn Hutter, who is a member of this organizing committee, and many others. And it's been a great thing for these people to come together and then involve many, many more people around the world to produce a white paper trying to describe the current state, the aspirations, and make recommendations about what we really should be focusing on to deliver on this dream that we can fully understand the genetic basis of common disease and go easily from maps to mechanisms to medicine. And I can commend you to the recommendations. There are different directions there and about 20 odd different recommendations. And so in the interest of time, I am going to stop there, knowing that I am just barely under the mark, and I am going to return back to Francis for whatever Francis would like to do, which is generally a good policy.
Well, I don't know about that part, but thank you, Eric. That was incredibly inspiring where we might be going with all of this. And we are at time, but I'm going to ask at least each of you to answer one question that I think might be in the minds of some of the people listening. In fact, came through in one of the emails that I received from somebody who's filtering this. People listening to these presentations, I think, are blown away by what's been accomplished, but especially by what now may become possible. And yet they may also be a little intimidated, feeling like, well, gosh, how can I be part of this if I'm not at the Broad Institute or I'm not working in Charles's lab or Claire's lab, but I want to be part of this opportunity for genomics to revolutionize everything we know about biology and medicine. So what would you say, and I'm particularly asking you, talk to the trainees. What would you say a trainee who's interested in incorporating some of this thinking into their own scientific pathway, their own career, what should they be doing right now other than going, wow, that was a great hour and a half?
Well, maybe I'll take a crack and then I'll pass to my colleagues here. More than ever right now, there are hundreds of things available to a trainee, even if you are not at a place generating data. By the way, don't imagine that you can't generate large amounts of data. Go do a single cell data set and you will suddenly have tens of thousands of cells, and that's an affordable experiment. And so please go make your own data and use it. But all these data on the web, I know from personal experience, living at a place where we generate a lot of data, that when we analyze it, we scratch about 1% of the surface of what can be learned. Each person who comes to these data sees different things. When we talk about the power of diversity in all its respects, the where you come from, the field you come from, the questions you care about, you have this vast thing and you bring a lens to it. I am confident, because I've seen it again and again, trainees everywhere will see something different in the data set. They will see hypotheses. They will be able often to test it on other data sets. They'll be able to generate data in their own labs. And the days when just a small number of people were generating the Human Genome Project data and gluing it together are long past. And one final point, one of the few good things about the pandemic is we've learned that where you are doesn't matter as much as we thought. And if you think you're not at a place that generates data, well, you're just not thinking virtually. I imagine that when we come back from this pandemic, certainly at the Broad, and I bet Charles and Claire will say the same thing and many others, I'm imagining we're going to set up networks of people who are at the Broad, even though they're not at the Broad. And I bet hundreds of other places will. So I think we'll probably blow up the model. For the trainees, you guys are so smart to have entered the field now, because now it's moving fast. All of us can tell you, how excited we were when we had the billion base bash in the Human Genome Project, and the whole community got together to celebrate 1 billion bases. And, I don't know, what is that, 3 minutes or something now? So you got born at the right time is all I can say.
Well said. So Claire, your perspective also here in terms of trainees, and maybe a bit about, are we democratizing the microbiome sufficiently? Is that something now that people have access to in a lab that doesn't happen to be yours?
Yeah, absolutely. I mean, Eric is a hard act to follow for anything, but all I can say is I enthusiastically echo everything that he has said. There is so much data available in public databases. One does not need to think that data generation needs to be a first step. I think what I will add that I didn't hear Eric say is that it is really important for trainees today to realize that the life sciences are very different than they are when all of us started. These have become quantitative sciences. So what you do need to do is make sure that you have the appropriate training so you know how to deal with large data sets, how to navigate these databases. That's something, again, if you can't do it where you are, there are so many resources available. You can take it upon yourself to make sure you have the relevant training. And with regard to your question about the microbiome, is it democratized enough? I would say yes, absolutely. To me, one of the things that it has just astounded me is how microbiome studies have infiltrated or percolated into almost every study section, if you will, at NIH. And I guess, if there was one thing that I would like to see different when a celebration is had in the next 10 years or 5 years or whatever the appropriate celebratory period is that we could hopefully have talks that are more integrated, because what we all spoke about is true in its own little universe. I shouldn't say universe, that's too pejorative. It's all true in its universe, but what we haven't yet done effectively is put human genome and microbiome studies together. Clearly, the first part of that is generating the data. I would love to see longitudinal microbiome data collected from all of the participants in the All of Us project. That's what we need to do. And then when we are at that point, we can really start to think about fully integrating all of the work that genomics has enabled. And I don't think until we do that, we're really going to have the full picture. The progress today was just mind-boggling. But I guess I would throw this out as a challenge to students and trainees who are thinking about coming into this field. There is so much more to be done, not only in these respective areas, but in bringing all of this information together.
Oh, that's very well said. And yes, I too dream of when all of us has this longitudinal microbiome added to everything else that's being collected about genomics and environment, electronic health records, wearable sensors, all the rest. And we need to get started with that. We probably can't do 1 million people every week quite yet, but that's the kind of goal that we ought to be aiming for. So Charles, you think about this a lot in terms of the democratizing that's necessary, not just in our country, but across the world. And you've done a lot with H3 Africa to try to tackle that in a very specific, practical way. You get the final word here. What would you want trainees to hear about who are trying to figure out, can they get engaged in this adventure?
Yes, they can. And I would seriously encourage our trainees to actually behave like a kid in the candy store, because of the excitement of genomics. Be bold in your questions that you're asking, and be ready to interrogate the whole genome in the questions that you are asking. And but I want to say again that yes, there are a lot of data sets that are available on the web, and but there are challenges for people from developing countries, scientists from developing countries, to interrogate those data because of poor infrastructure. Mm-hmm. So I think we need to be developing technology and ways to make those data available to individuals who may not have the best internet connection to be able to interrogate this data. And one of the ways we are doing that in terms of H3 Africa is to begin to encourage the development of bioinformatics network that individuals can have local access to in a way that they can actually begin to enjoy this wonderful precedent that genomics has shared, and that is data sharing. And the fact that we encourage each other to put our data up. But if we are not able to analyze it because of limitation of infrastructure, then we have a problem on our hands. So I really encourage our global colleagues to think about this data, to use this data, but to begin to think of a way to solve local problems that may be a hindrance to their ability to get access to this type of data.
Well said. Well, I know we've run a little past the time that people had allotted for this, but I didn't want to have us close down without at least having a little bit of a conversation here. And this is a very relevant one indeed. So I want to thank all three of our wonderful speakers for giving us this kind of perspective, both about how we got here and particularly about where we are going. And yeah, Claire, I'm not sure what the right interval is for the next time we have another symposium celebrating anniversaries, because we have enough things we could do this every couple of months, but probably that would get a little tiresome for everyone. But I am quite sure if we gathered together in 5 years, that there would be things we would talk about then that we didn't even know were coming today, because the pace of development of technologies and insights has gotten to be so rapid, almost exponential. So if people are interested in joining this adventure, there's a lot of fun to be had. And I agree with what Eric said, that the generation that's just now jumping in to the scientific community and biomedicine is like the luckiest generation yet, because so many questions are going to be answerable that we hardly even know how to ask before. And now we're going to get those solutions. So, and genomics will not be the only way to get there. Let's be clear. We're not being so narrow here. I'm the NIH director. I really love all of the biomedical research landscape, which is massive and amazing and wonderful. But genomics does tend to be kind of in the center of a lot of this. And it also is empowered now by the kind of technology that has emerged in the last 20 or 30 years that we can take full advantage of to really push back the frontiers of our ignorance. So thank you, everybody, for listening. Thank you, speakers, for giving us such an exciting set of presentations. Thank you, Wednesday afternoon, for having us all come and join in this symposium at NIH. With that, I wish you all the best.
Bye.
Farewell.
Bye.